Heart disease. In developed countries, it is the most common way to die. Why? Well, soon after we are born, our hearts almost completely lose the ability to heal. So, if we slightly injure our hearts and a few cells die, the remainder of the heart is more stressed out. This causes more cells to die, and this increase in death causes more stress. This vicious circle continues until, inevitably, over the course of a few years and decades, if another disease doesn't get you first, your heart fails and you die. Modern cardiovascular medicine can slow this process down, but it cannot stop it and it cannot reverse it. But why can our hearts heal in the first place? Well, a big reason is because our hearts are muscles, and the cells of our heart, the muscle cells of our heart, or cardiomyocytes, like other muscle cells, have difficulty with cell division. The skeletal muscle cells in the rest of your body can't undergo cell division at all. They have many nuclei, and their cytoskeleton is completely locked up in muscle fibrils. This prevents them from forming mitotic spindles, and they can't divide. But after a workout, your muscles don't have difficulty regenerating. In skeletal muscle, there are stem cells called satellite cells that proliferate and fuse together to form new muscle fibers. But, as we figured out after decades of searching, our hearts do not have a satellite stem cell system. Instead, in those organisms whose hearts can regenerate, like adult zebrafish or baby mice, what happens is that existing cardiomyocytes, some of them will lose their muscular structure and form mitotic spindles. They will undergo a few rounds of cell division, and then their daughters will mature back into full muscular beating cardiomyocytes. But for some reason, in mammals, our cardiomyocytes lose this ability with age. We're not entirely sure why, but there's something that goes on in the cytoskeleton that appears to be a major player. In a normal cell, you have a centrosome surrounded by paracentriolar material out of which the microtubules emanate. This is also the case in young cardiomyocytes that can still undergo cell division. When a normal cell or a young cardiomyocyte divides, the centrosomes duplicate and send out their microtubules to the center to overlap and grab onto the chromosomes. Where the microtubules overlap, or the equatorial band, the proteins like aurora kinase B will be activated, and these proteins will work together to form the cleavage furrow and pinch the cell in two. But as cardiomyocytes age, they lose their centrosomes, and their paracentriolar material and microtubules begin to emanate out of the nucleus instead. When a cell like this attempts to divide, a neat mitotic spindle does not form. There is no neat equatorial band, and there's no neat equatorial band of aurora kinase B expression, and thus there's no organized cleavage furrow. When you record these cells in a petri dish trying to divide, they'll form an incomplete cleavage furrow, try, and then just kind of give up, pop back into their original shape, and end up with a large cell with two nuclei instead. By the time you're an adult, the majority of your heart cells are binucleated. But this isn't the end of the story. We have in fact found ways of making adult cardiomyocytes in at least rats and mice divide. The thing is, we don't really know how these treatments work. The ones that we've studied so far are tumor suppression gene knockouts like P21 and P27, or by knocking down the hippo organ size control pathway. Both of these seem to cause many new cardiomyocytes to be born in adult mice and rats, and lineage tracing experiments show that these cells were born out of existing older cardiomyocytes. My hypothesis is that part of the mechanism by how these treatments work is that they induce adult cardiomyocytes to reform their centrosomes so they can reform proper mitotic spindles and thus divide properly. How I will do this is by disassembling adult rat hearts in petri dishes and staining them with a microtubule stain sertubulin and the actin stain LifeAct. I will record these cells under a fluorescent microscope with or without the cell division inducing treatments, and I expect that some of the cells exposed to the cell division inducing treatment will be observed to reform centrosomes and reform mitotic spindles. If my hypothesis is true, I would then, as a follow-up experiment, expect that proteins involved in the assembly of centrosomes, like paracentrin, are upregulated in cells that have been treated with cell division inducing treatments. With this, our lab or other labs could follow up and finally find a mechanism by which the gene expression changes caused by the cell division treatments result in a rearrangement of the cytoskeleton. And thus, we would finally have a little bit of the mechanism of how adult cardiomyocytes are induced to divide. And we'd be one step closer to porting such a treatment to humans and get a little bit closer to finally healing our hearts.